Hi and welcome to QSD. Tell me, have you any experience of Alzheimer's? If you have, you probably hate it. It robs the older generation of their treasure trove of memories. But what causes it? Well, we may have an alternate contender if this research is anything to go by. So keep watching to find out more, and at the end I will try to see the bigger picture. Alzheimer's disease then. This is the most common cause of dementia among the elderly and is characterised by plaques and tangles in the brain. Plaques are deposits of a protein fragment called beta amyloid, and they look like clumps in the spaces between neurons. Tangles are twisted fibres of tau, another protein, and look like bundles of fibres that build up inside cells. However, in addition to plaques, lysosomal storage is observed in brains of people who have Alzheimer's disease. This area serves as the cell's recycling centre where old proteins and lipids are broken down to their building blocks before being shipped back to the cell to be repurposed into new proteins and lipids. To maintain functionality, the synthesis of proteins is balanced by the degradation of proteins. But if what enters does not get broken down into little pieces, then those pieces also can't leave the lysosome. The cell decides it's not working and it stores it, meaning it just pushes it to one side and starts to make another one. And if that one has the same problem, then it pushes it to one side and starts to make another one. And the process is repeated over and over again. Now take it, you can probably see how this could cause a problem. Well, let's say neurons. They're fragile cells that cannot undergo cell division, and these are particularly susceptible to this type of damage. The standard type of lysosomal damage that shows up weeks after birth and is usually fatal within a few years. But with Alzheimer's, the same thing shows up much later and the progress is really slow in comparison. But if we take an analogy like bleeding, just because an artery bleeds faster than the vein does not mean that one should be treated and the other shouldn't, or they are radically different. Researchers in the Department of Chemistry and the Division of Biomedical Sciences at UC Riverside propose that long-lived proteins can undergo spontaneous modifications that can make them undigestible by the lysosomes. The results of their recent study appeared in a journal of the American Chemical Society just recently. It appears that changes occur in the fundamental structure of the amino acids that make up the proteins, with amino acids spontaneously acquiring the mirror image of their original structures. This then stopped the necessary enzymes from breaking down the protein because they were unable to latch onto it. This paper shows that this structural modification can happen in both beta amyloid and tau, both proteins relevant to Alzheimer's disease. They also show that this is a function of time. So the older it is, the more likely it is they will go rogue, as it were. And furthermore, when these proteins undergo this change, it is almost invisible, and this may well explain why researchers have not paid attention to it previously. If this is correct, it may well open up new avenues of treatment and ultimately for prevention of this disease ever taking hold. The findings could also have implications for other age-related diseases such as macular degeneration and cardiac diseases, all of which are linked to lysosomal pathology. Ryan Julian added that it's long been known that these modifications happen in long-lived proteins, but no one has ever looked at whether these modifications could prevent the lysosomes from being able to break down the proteins. One way to prevent this would be to recycle the proteins so they are not sitting around long enough to go through the whole process. But currently there are no drugs available to stimulate this recycling, a process called autophagy, for Alzheimer's disease treatment. Well, that's all fine and dandy, but how does that all fit into the bigger picture? Let me start by asking you this. What if you didn't age? You kept the same muscle mass, the same reaction time, the same cardiac ability, the same perfect eyesight, crystal hearing. Well, you get the picture. Turns out the things we thought were inevitable aging are actually just a basic biological process that's not being done as efficiently as it was a few decades ago when you were younger. Unless you're not past your 20s yet, that is. One of those processes is autophagy. The ability of the body to cannibalise old useless material and to recycle it again for reuse, and with anything unusable and unwanted being disposed of. But as things that cannot be broken down build up and the process stops running as efficiently, backlogs build up and you experience the process we call ageing. Externally, you may see wrinkles, loose skin, lost muscle mass and tone, and internally it's much the same. But what if you could restore that functionality back to peak performance? Well, there are clinical trials currently underway for treatments that do just that. Or alternatively, there is evidence starting to show up of how fasting may stimulate some of the same processes. But that is maybe for a future episode. So, what do you think? Am I right or am I wrong? Do you want to live forever? If not, how do you want to die? Tell all and cite evidence where required. You know the score. Thanks a lot and see you soon.